First, what do we mean by logic circuits? In computers and electronics, logic circuits are tiny networks of switches, transistors, that perform basic operations like and, or, and not to process information. They are the foundation of all digital technology. Everything from calculators to smartphones depends on them. Now imagine if we could build similar logical operations inside living cells, not using silicon or wires, but using biological molecules. This is exactly what researchers are trying to achieve with protein-based logic circuits in synthetic cells. But what is a synthetic cell? A synthetic cell is an artificial or engineered cell-like structure that mimics certain functions of natural cells. Scientists build these to study lifelike behaviors, create new therapies, or design tiny biological machines. So, how can proteins act like logic gates? In biology, proteins do not work alone. They often interact with other proteins in very specific ways. For example, one protein might activate another or block its action, depending on the situation. By carefully designing these interactions, scientists can make proteins perform logic-like behaviors. Imagine two proteins, A and B. Suppose A can only activate a certain reaction if B is also present. This is similar to an AND gate in electronics. Both inputs must be on to get an on output. Or suppose a protein acts only if another protein is not present. That's like a NOT gate. By combining these types of interactions, you can create more complex logical decisions inside a cell. You might ask, why would we want to build logic circuits in cells? Good question. There are many exciting reasons. Smart drug delivery. Synthetic cells with logic circuits could decide when and where to release a drug based on signals from the environment or from the body. Biosensing. Cells could detect combinations of chemicals or disease markers and produce a response only when certain conditions are met. Controlling gene expression. Logic circuits can turn genes on or off precisely, which is useful in synthetic biology and advanced gene therapies. A real example, researchers have designed synthetic circuits where proteins act as sensors that recognize certain molecules and then decide whether to produce a fluorescent signal. If molecule X and molecule Y are both present, the cell lights up. This is exactly like an AND gate, but inside a living system. Another question you might ask, how do proteins know how to interact correctly without causing chaos? This is where careful design comes in. Scientists use detailed knowledge of protein structures and interaction patterns to design new proteins that fit together only in specific ways. Think of it like making a custom puzzle where each piece fits only in one spot. Unlike electronic circuits, protein-based logic circuits are flexible and can be reprogrammed by changing the protein sequences. However, they are also slower and more complex to control because they involve biological processes, not fast electrical signals. In short, protein-based logic circuits in synthetic cells show us a new way to bring computing into the realm of biology. Instead of using wires and electricity, we use proteins and molecular interactions. This approach opens the door to new smart therapies, advanced diagnostics, and artificial biological systems that can think and respond to their environment in very precise ways. Imagine a future where tiny, programmable cells roam your body, making real-time decisions to keep you healthy. All thanks to logic circuits made not of metal, but of proteins. Biological systems inherently process information through molecular interactions and signaling pathways. Traditional synthetic biology has focused on genetic circuits using DNA and RNA to program cellular behaviors. However, these circuits often suffer from slow response times and require transcription and translation, limiting real-time control. Protein-based circuits bypass these delays by leveraging rapid, direct protein-protein interactions for computation. Using proteins to perform logical operations inside cells opens up possibilities for fast, dynamic, and highly tunable cellular programming. Researchers have designed protein networks that quickly respond to external stimuli and activate downstream signaling within seconds. Like using electrical switches instead of writing software code, protein logic circuits allow immediate, direct control inside cells. Logic gates are the fundamental building blocks of digital computation, performing operations such as and, or, and not. In protein-based systems, these gates are built from engineered protein domains that bind or dissociate in response to specific inputs. By controlling conformational changes or assembly/disassembly of complexes, proteins can produce binary outputs, on or off states. These designs exploit modular protein-protein interaction motifs, allowing combinatorial logic functions to be encoded in living cells. The resulting circuits can integrate multiple signals and make cellular decisions based on complex input combinations. A designed protein AND gate activates only when two specific signaling proteins are present simultaneously. Like requiring two keys to open a safe, an AND gate protein needs two binding partners to activate a cellular response. Protein-based logic circuits rely on precise engineering of interaction interfaces such as coiled coil domains, SH2 domains, or PDZ domains. These modules can be designed to bind selectively to one another under specific conditions, allowing modular construction of logic networks. Mutagenesis and computational protein design enable fine-tuning of affinity and specificity to minimize unintended crosstalk. The dynamic and reversible nature of protein interaction supports rapid switching and flexible reconfiguration. This engineering toolkit empowers synthetic biologists to build increasingly sophisticated and reliable logic systems inside cells. Engineered coiled coil pairs have been used to create programmable protein switches controlling cellular fluorescence. Like designing custom Velcro strips that only stick to a unique counterpart, interaction modules provide selective connectivity. Synthetic cells are minimal engineered cellular systems designed to mimic certain functions of living organisms. Incorporating protein-based logic circuits into synthetic cells allows controlled, programmable responses to environmental or internal stimuli. These circuits can regulate synthetic cell behaviors such as motility, division, or metabolite production without relying on gene expression. The self-contained nature of synthetic cells provides a safe platform for studying and deploying protein logic systems. Such integration paves the way for creating artificial cells with life-like decision-making abilities. A synthetic cell programmed with a protein circuit can move toward a nutrient source only when two metabolic markers are detected. Like a robot programmed to move only if both light and heat sensors are triggered, synthetic cells follow precise protein logic instructions. Protein-based logic circuits respond significantly faster than genetic circuits, since they avoid transcription and translation delays. They operate at post-translational levels, allowing near-instantaneous signal processing. Additionally, they reduce the cellular burden associated with producing and degrading RNAs and proteins for genetic control. This minimizes metabolic disturbances and enhances the stability of engineered functions over time. These advantages make protein logic ideal for applications requiring rapid and reversible responses. Protein-based toggle switches can flip states within seconds, compared to several hours needed for gene-based switches. Like flicking a light switch instead of waiting for a sunrise, protein circuits offer immediate control. Designing highly specific protein interactions without unintended cross-reactions remains challenging. Cellular environments are crowded and dynamic, introducing noise that can interfere with circuit fidelity. Maintaining stability and preventing degradation of engineered proteins over time also require careful optimization. Furthermore, scaling up complexity, e.g., building large networks with many logic gates, increases design difficulty and error risk. Overcoming these obstacles is essential for broader adoption and practical deployment in therapeutic or industrial contexts. Early protein circuits sometimes triggered unintended pathways due to nonspecific binding in crowded cytoplasmic conditions. Like building a house of cards in a windy room, precision and environmental control are crucial for stability. Protein-based logic circuits can be harnessed to create smart biosensors capable of integrating multiple biomarkers before triggering a response. In therapeutic contexts, such circuits enable targeted actions, such as releasing drugs only in the presence of specific disease markers. This approach reduces side effects and enhances treatment precision. In diagnostics, protein circuits can perform on-the-spot logical analyses of patient samples. These applications demonstrate the transformative potential of protein-based computing within living systems. A therapeutic protein circuit can selectively activate an apoptosis signal only when cancer-specific proteins are detected inside cells, like a safety system that deploys airbags only if multiple crash sensors are simultaneously triggered. Future work focuses on expanding the library of programmable protein modules and improving predictive design algorithms. Integration with other molecular layers such as RNA-based sensors or small molecule signals can create multimodal synthetic networks. Advances in directed evolution and AI-driven protein modeling promise more robust and versatile logic components. Developing standard frameworks for assembling and testing these circuits will accelerate translation into practical applications. Ultimately, this field aims to create fully autonomous cellular systems capable of complex decision-making and adaptive behaviors. Researchers are working on protein circuits that integrate metabolic and mechanical signals to control synthetic tissue growth. Like upgrading from simple calculator functions to a programmable computer, these advances expand the possibilities of cellular thinking. The ability to program cells with protein logic circuits challenges the traditional view of life as purely gene-driven. It suggests that cells can be redefined as programmable machines capable of processing information through entirely synthetic, non-genetic networks. This perspective blurs boundaries between biological and artificial systems, raising ethical and philosophical questions about cellular autonomy. In practical terms, it enables the design of highly controlled biological devices for medicine, industry, and environmental applications. Exploring these systems forces us to reconsider what defines intelligence and decision-making in living systems. Synthetic cells equipped with protein logic circuits can autonomously adapt to changing environments, resembling primitive artificial intelligence. Like teaching a puppet to move independently without strings, programming cells with protein circuits gives them a new, unexpected agency.